I'm our programs coordinator here at Family Sciences Center. You can find me in the whale lab. I think I'll be seeing at least one course pretty consistently in the next 2 weeks. You're going to be happy because I love the company in there. It's an awesome place. It's so great to see you, and we're so excited you're here. This evening, we are so thrilled to welcome Dr. Dina with us here tonight. Our talk is going to be absolutely fabulous. Dina came out here in 2019 and also got to spend about 18 months here as a postdoc here at BMSC. And we're so happy that you're back and so grateful that you've generously offered your knowledge and your time this evening. And we can't wait to hear from our speakers. So please welcome.
Thank you. Can everyone hear me? Is this doing anything? I'm not sure. All right, perfect. So I am a science communicator first and foremost, so I kind of like to know my audience. I know we have at least one undergrad in the room. Are all of you undergrads? Do we have some grad students? Okay, so we've got some undergrads, we've got some grad students. All right, great, excellent. So super excited to be here, super excited that part of my job is bringing me back here year after year. It's a really special place, as you guys will learn or maybe already know. I like to think the hardest part of organizing these talks these days is now fitting all of my collaborators and institutions onto the title slide, so it's all downhill from there. Anyways, the big picture of where I want to go both with this talk and sort of in my career overall is all about plasticity. So what is plasticity? Plasticity is the ability of a single individual with its single genome and genotype to produce multiple different phenotypes in response to different environmental cues. I find this easiest to grasp with an example. So the Daphnia that I'm showing you there, they're not just fish food, although for the stickle nerds in the room, maybe they are just fish food, but they, they are kind of interesting in their own right. Because these two individuals are actually genetically identical to one another. So they undergo clonal reproduction, and these two, despite their phenotypic differences, are in fact in possession of identical genomes. And so the difference between them obviously is the helmet on the one on the left and the longer tail spine, and then the one on the right doesn't have the helmet and has a much shorter spine. And that's just based on the presence or absence of predators in their environment. Now, the benefit to growing spines, I think, when you're actively experiencing predation is fairly self-explanatory. You look bigger, scarier, meaner to your predators and might be able to avoid predation entirely. Or if you do start to be predated, you can kind of hurt them and in the process of hurting them, startle them and get away. But there's also an associated cost to growing those spines, which is in the resources that they take away from other vital functions, including reproduction. So in a lot of ways, it makes sense that you would just want to grow those spines in the context where they're actually going to be beneficial, where they're going to be helpful in escaping that predation. So I would say that the plasticity here is adaptive, right? You can express the trait in the context where it's going to help you out, and you can save on those resources when it's not going to be necessary to spend them. And we do find examples of plasticity and adaptive plasticity all across the tree of life, including these spadefoot toad tadpoles up at the top left. They have omnivorous and carnivorous forms, which just allows them to escape competition from their siblings and cousins and everybody else who lives in the same pond as them. We also see this in fish. So these fish pharyngeal jaws, so that's the jaws at the back of their throat that help them to actually process their prey. These are taken from individuals that either specialized on eating snails or eating vegetative matter. And I think these examples might give you the false impression that plasticity is so amazing and so wonderful and so adaptive that it's ubiquitous, right? Everybody should be plastic all the time. But that's not actually the case. And the reason for that, we think, is that plasticity itself comes with an associated cost. And this leads to a situation where we have closely related species that differ in both the amount of plasticity they show, so how severe of a response they show, as well as, in some cases, the direction of that response as well. And so we can measure those differences using these tools called reaction norms. So species 1 here is showing a low trait value in the first environment, but a higher trait value in the second environment. So I would say it's pretty plastic. Species 2 here isn't showing much of a difference in its environments, and so it's not very plastic. Species 3, of course, is plastic, but in the opposite direction from species 1. Now, this is a hypothetical example of made-up data, obviously, but we can do this, and I have, in fact, gotten a pattern, as you will see, that's pretty similar to this in real life. But the key point I want to make here is that the difference in both the pattern, so the direction, and the magnitude or the amount, the severity of the response in plasticity means that plasticity is probably a trait that itself can evolve. And if it can evolve, there should be some genetic basis that we can find for it. But that genetic basis, the genes and the signaling cascades that enable this ability, are not particularly well understood, especially not in vertebrate systems. So my past work tried to fill that gap. And I was focusing in on the benthopelagic axis, which is just sort of describing where fish tend to live and feed. Pelagic feeders, those are the ones that are up in the open water column, so they're suction or ram feeding. Benthic feeders are pretty variable, but they all are sort of have the shared trait of directly contacting the substrate. So they're biting, they're scraping, they're sifting or plucking their food directly off the bottom of their environment. And so they're making that direct contact. I will also put the caveat out there that I use the terms pelagic and limnetic interchangeably. There is probably a limnologist out there who's coming to get me for that one of these days. But for me, I'm just using the term to describe where they're living and feeding, and if they're living and feeding up in the open water column. And we find benthic and pelagic specialist species in lots of different fish taxa. So what I'm showing you right now are all cichlids, but we can find similar examples in ice fish and sunfish, and of course in the three-spined stickleback. So there is an interesting thing going on here, which is that the benthic fish tend to share some traits with the other benthic fish, even though they're in completely different taxa and aren't as closely related to each other as they are to their pelagic counterparts, right? The stickleback is more closely related to the stickleback, the cichlid is more closely related to the cichlid, but the benthic cichlid and the benthic stickleback have these traits in common in terms of having shorter overall jaws, deeper bodies, steeper craniofacial profiles. And what I mean by that is really just like, if you imagine walking up the hill of the face, is it super steep and you don't want to walk up that hill? Or is it much more shallow and it's a really easy walk? So we have those traits. We also have some changes to the fins as well. And these differences are due in part to the unique challenges that they're experiencing in these different dietary feeding modes. One of those challenges that I can use this slide to sort of demonstrate is differential loading. So I think it's fairly easy to see the benthic fish, especially this guy, right? He's loading his face because he is pushing his face right up against the substrate. And he has to put quite a bit of mechanical force into that interaction. Now, the pelagic fish, it's easy to say, well, he's not loading his face. He's not pushing up against anything, right? That's what I thought when I started this project. But actually, the pelagic fish is also, I would argue, loading its face. It's just doing so in a very fundamentally different way. Pelagic fish, they rely on the rapid and repeated opening and closing of their jaws as they're making this feeding motion. And so that puts a high cyclical load, or the sort of motion of the repeated motion is what's causing the load there, as opposed to that direct contact with the substrate. So different loads, both loading just in different ways. But what we see in these images is a pattern of trait covariation across wide timescales, right? The benthic fish looks like the benthic fish, even though they're not very closely related. And for me, that always begged the question, is this genetics? Is it plasticity? Is it some combination or interaction between the two? And hopefully I'll convince you tonight that it's both, realistically speaking. So to do that, I'm going to tell you two different vignettes focusing on three different systems because I'm insane. I started out with cichlids and zebrafish, so that's the story I'm going to tell you first, and then we'll move on to talking a little bit about stickleback and what I want to do with that system moving forward. And we see plasticity along this axis in lots and lots of taxa, including cichlids. So fish tend to be behaviorally generalist. What this means is that they exhibit plasticity in their behavior. They can move from feeding on one way to feeding in the other way, depending on what's available to them in their environment. And lots of cichlid species are plastic in this way, which is one of the reasons why we used them. So I'm going to show you two videos of the same species different individuals, but the same species feeding in these two different ways. The pelagic fish, we ground up their food and just sprinkled it into the water column, so they had to suction it out of the water column just as they would if they were pelagic in nature. The benthic fish, we took the food, we mixed it with an agar solution to make it sticky. We spread it over the lava rocks, we let that dry completely, and then we sank it to the bottom of the tanks, forcing them to scrape. And what you will hopefully appreciate, if I can get these videos to play, is that they do this pretty well in either, oops, they do this pretty well. Sorry. Unfortunately, I'm not getting my technology to work today. In any event, we can get them to feed in different ways, and we can get multiple species to feed in different ways. So we can get Trophiops redcheek, which is the one that I'm showing you right here. We can also get Labiotrophus fleurborni, or LF, and Metriaclima zebra, or MZ, all from the same lake. They all exhibit this kind of behavioral plasticity. But when it comes to the morphological plasticity, it's actually quite a different story. So Trophiops redcheek, which is the species from the videos, this is the same fish after about 5 months of diet treatments. And what we found was that they were exhibiting steeper craniofacial profiles, deeper overall heads, and shorter jaws relative to their body size compared to their pelagic counterparts. So that's all exactly in line with what we would expect for a benthic versus a pelagic feeder. When we looked at a different species, this is Labiotrophus fleurborni, or LF, they also show that behavioral plasticity, but they don't show that morphological plasticity. There's really not much changing between the benthic and the pelagic environments here. And again, for me, this begs the question, what are the genes that are allowing TRC to respond plastically in both their behavior and their anatomy, but are not allowing LF to do the same thing? So fortunately for us, my PhD supervisor and my PhD, these two species will hybridize readily in the lab. So that lets us use a QTL mapping cross to try and answer this question. For those of you who aren't familiar with QTL, basically all it does is it takes advantage of the fact that when you make hybrids of two different individuals, you mix up their gene pool. And you do this with recombination in multiple generations. And you can try to associate the variation in a trait with its underlying genetic causes. So in this case, we were doing this using the magnitude of their plastic response as our trait of interest, allowing us to hopefully map the genetic basis of plasticity. Happy to talk your ear off about stats if you want to know how that happened. But one of the genes that we identified through that QTL analysis was patch1, and specifically for craniofacial variation, craniofacial plasticity. And patch1 was particularly exciting to us, partly because it's a key receptor in the hedgehog signaling pathway, which is a hugely important developmental pathway that is also known for influencing craniofacial shape in lots of different systems. And we knew that it was doing that in cichlids specifically because we manipulated hedgehog signaling with a known small molecule inhibitor, which is cyclopamine. And we showed that the shape of one of the bones in the face was actually responding to the cyclopamine treatment. So LF has a particular shape. LF with the carrier control has a very similar shape. When you inhibit hedgehog signaling, then things change so that they start to mimic one of our other species. All well and good, showing us that hedgehog signaling is likely important to craniofacial variation and divergence in cichlids. Great. But we were also excited about patch1 as a potential plasticity gene because it is associated with this developmental behavior that I'm showing you on the slides here, which is the rapid opening and closing of their jaws over and over and over again throughout development. And so why are these fish doing this? That was the question that my lab mate Enon asked of my PhD supervisor, and Craig sort of responded, waving him off like, yeah, they're breathing, whatever. And that didn't sit super well with Enon. So we looked into it a little bit further. And as it turns out, and you can kind of see why this might be the case, if you look at where their gills should be, there's really nothing going on quite yet. But if you look at the skin over their yolk sac, there's tons of blood vessels, and as it turns out, gas exchange at this stage of their development is mostly happening through their through the epidermal cells on their yolk sac, and not through the gills, which aren't yet developed. So, Enon asked again, why are they doing this? They're spending a ton of energy to perform this behavior over and over and over again. And as it turns out, the answer he arrived at was that maybe it's mimicking that cyclic load that I was talking about earlier in the talk, where they're actually working out their jaws in preparation for becoming adults and doing the adult behaviors that they're going to have to do. We have some strong evidence in support of that, including the fact that species exhibit this behavior at different frequencies from each other, and that if you force them to change their frequency, it does in fact change the shape of some of their bones in their face. And, ooh, not sure what that was. These differences all also coincide with differential patch1 expression. So the data here on this slide are all kind of pointing us to hedgehog signaling, and maybe patch1 in particular being a key mediator of jaw development being plastic. And we wanted to sort of try to link those two things together and decide if, you know, hedgehog signaling is also involved in specifically that plastic response. So to test that hypothesis, we needed a faster and easier way to assay plasticity. And what we arrived at as a good tool to do this was bone deposition. So just sort of measuring how quickly they're depositing bone. Or how much bone they're depositing in the same amount of time is really how I should say that. So we labeled the bone with a fluorescent marker at the beginning of the experiment. We labeled the bone with a fluorescent marker at the end of the experiment. We let them be eating in their different diet treatments throughout the experiment, and then we could simply measure how much bone was deposited based on the space between the two labels. So the data that I'm showing you here are from one of the important bones in our highly plastic species, TRC. And you can see that it does exhibit more bone deposition in the benthic treatment than in the pelagic treatment. We can see this visually. Those two specimens that I'm showing you are size matched. And we can also see this graphically, and it was borne out statistically in sort of the average bone deposition in each of these treatments. So we have that nice steeply sloped reaction norm there. When we looked at MZ, which was our third species that we were looking at, they share a patch1 allele with TRC. So the expectation naively was that they would show basically the same plastic response. And to a degree, yes, and to a degree, no. There's a similar magnitude of plasticity, so the slope of the reaction norm is approximately similar across these two species. MZ seems to have higher individual variation, as evidenced by the width of the confidence intervals there. But of course, they also have an opposite direction where MZ is depositing more bone in the pelagic than in the benthic treatment. We can talk about why later if there's time, so save that question if you're interested. But when we looked at LF, which was plastic behaviorally, but not so much morphologically, has a different patch1 allele from either TRC or MZ, we expected there wouldn't be a plastic response, and hey presto, that's exactly what we saw. And as I mentioned earlier, this does really nicely mimic that sort of textbook hypothetical example that we had been using in the intro for this project for years before we ever got that data, so we were super stoked about that. And I will also point out that I'm showing you the data from a single bone in their face, but I measured about 8 different bones in their face, and they all showed basically the same thing. So we were feeling pretty confident that we had the sort of tissue level mechanism of plasticity in terms of this differential bone expression, and that would work really well for being able to assay plasticity moving forward into the future. We did also do some qPCR work on these guys and showed that there was a correlation between the increased bone deposition and the increased patch1 expression. In other words, TRC had higher patch1 expression in the benthic versus the pelagic treatment, and MZ it was actually flipped. So we did show that response. I would love to be able to tell you that we looked at LF and there was no significant difference. We had run out of LF individuals by that point and we weren't able to find any more. So we don't actually have that data, but my strong suspicion based on this is that it would hold true. But I promised that I was going to try and test patch1 as the causative gene underlying plasticity in the face. And so for that, we turned to zebrafish. Zebrafish do exhibit behavioral plasticity in much the same way that cichlids do. Again, this is pretty impressive for something that doesn't have, I'll show you just the benthic again. For something that doesn't have teeth, the fact that we can force them to scrape food off of a tile is pretty crazy. Did not expect that to work as well as it did. But they do exhibit this behavioral plasticity. And we were curious to see if they would also exhibit morphological plasticity. And so my second postdoc actually tried to answer that question, looking at the response of body shape to these dietary shifts. One of the frequent pieces of critiques that I got from my reviewers of my papers was that we didn't know what they looked like before we put them in the treatments. They could have started out in a different place, and that is technically true. So I imaged these fish as I was putting them into their treatments, did the exact same analyses that I'd been doing in the past, and showed that they stacked up on top of each other pretty well. And I will say the control was just their normal GM 300 zebrafish food. The pelagic treatment was the same food ground up, and then the benthic treatment was the food mixed with agar onto the tiles like I showed you in the video. So there were no differences between the treatments. That was statistically borne out as well. There was no significant difference in those shapes. And when we look at what the average quote unquote fish in this analysis looked like, it looked pretty much like, you know, a regular zebrafish. What I did next was we ran the exact same analyses on the exact same fish 5 months later. And what we see is that the pelagic and the control are still stacking up fairly well. The benthic fish are now starting to do something quite different. That difference was significant according to an ANOVA, and the post hoc test that we did confirmed that it was what we would expect from the visual analysis, that it was the benthics that were doing something substantially different from the controls and the pelagics. And again, that kind of makes sense because the pelagic and the control diet was quite similar, whereas the benthic was sort of the odd man out there. So here's what those treatments look like. And again, the shapes represent the shape of a fish that exists in the part of the PC plot according to the letter. So your B is for your benthic, and your C is for your control and pelagic. And we're seeing some clear differences again in the steepness of the craniofacial profile, the body depth, the pectoral and anal fin placements, all the things that we would expect from these different diets. So we switched over to using zebrafish for these kinds of analyses because we had pre-existing transgenic lines. I didn't have to make them. They were already, in fact, one door over from where I already worked, so I didn't even have to go very far for them. We had these pre-existing transgenic lines where we could actually increase or decrease hedgehog signaling using heat shock. So that allowed us to do it in a really time-specific manner, which is important because if you tinker with hedgehog signaling too much early in development, they die and you don't get to do the beautiful assays that you want to do with them. So we waited for them to grow up. We hit them with the heat shock. That was able to turn up and turn down this signaling pathway. So the data that I'm showing you first here is from the wild type. There is clear plasticity in their bone deposition, where the pelagic fish are depositing more bone than the benthic fish. If our hypothesis is true and hedgehog signaling mediates plasticity, if we decrease hedgehog signaling, we expect that reaction norm to flatten out, to look more like this. If we increase hedgehog signaling, we would expect that reaction norm to get steeper. What did we find? Well, that's our hedgehog downline, looking pretty flat, much less dramatic. Here's our hedgehog upline, also showing us exactly what we expected to see. That never happens. I don't know what happened here, to be honest with you. But what was neat about this is that we were able to show both loss and gain of function of this gene by tinkering with this gene. And that's kind of neat as well because gain of function is usually a little bit harder to find. So it was pretty stoked that this was our results here. That said, there were still some open questions, including what other genes are involved in zebrafish plasticity, because initially we were just doing this because we couldn't do it in cichlids as easily or as quickly. We didn't really know very much about what that plastic response looked like in zebrafish because they were zebrafish. They're a lab rat, right? They're not supposed to have this crazy natural variation. So no one was really looking at that. which is where my postdoc with Tetsuya Nakamura came in. We were subjecting different fish to the same diet treatments for 0, 4, and 14 days. I dissected off 3 tissues, their pectoral fins, their faces, including the interopercle, which is one of the bones I've been showing you this whole time, and the shoulder girdle. And I took those tissues at every one of those time points. Then we used the dissected tissue for an RNA-seq analysis and started running a bunch of differential expression analyses. I started with the pectoral fins because those were my postdoc supervisor's main interest. And so this is the differentially expressed genes at day 4. There were some interesting genes popping up here, including TRIM63, which is a muscle ring protein. And given the interaction between bone and muscle, that made some sense that it would be involved in plasticity in these fish. So definitely want to follow up on that one. We also had some interesting results in the day 14 pectoral fins. So here, EPX, it's an inflammatory response gene. It can also be involved in cartilage and muscle formation, so that one's also particularly intriguing. And we've got this figure gives me nightmares, but we've got tons and tons of differentially expressed genes in their faces as well. But here too, a TRIM muscle protein is popping up. Recently, my collaborators, Tom Stewart and Aisha Iqbal at Penn State, they carried out some gene ontology analyses of the RNA-seq data. They were looking for transcription factors that were differentially expressed in our treatments versus our controls, and there were 98 of those. They started looking into some of the themes of those genes, and this really broke down into about 4 different themes. Genes that are involved in DNA methylation, including 2 different DNA methyltransferases, histone modifications, chromatin remodeling, and also fin development. So all things that were making sense with respect to what we were actually looking at, especially if we think about how the changes in chromatin conformation, whether it's tightly or loosely wound, those can be changed on a dime in response to environmental cues. And so this does make some sense that DNA methyltransferases might be involved here. We also did ATAC-seq. So we were looking for differentially accessible chromatin. And we're still actively analyzing that data, but one key finding so far is that DNMT1 is still present in this analysis as well. So that's a really good, juicy one for us to go after first, because it's both differentially expressed and differentially accessible. The 2 different lines of evidence are agreeing with and supporting each other that this might be a really important one. And of course, it has a reasonable function for what it might be doing in the system. So if I had to distill that first vignette down to a single slide, it would be this one, which is that we found the genetic basis of plasticity in cichlids, and we are working and continuing to uncover that genetic basis in zebrafish as well. But as I was starting my independent research at UFE, I really wanted to know if sticklebacks show the same kind of responses. And if so, what the genetic basis of those responses looks like relative to these other systems. Is it hedgehog signaling again? Is it something else? So that's the big picture of where I want to go with my independent research program, but I'm not quite funded for it yet. So I have been doing work on stickleback in the meantime. That's what brings me back here. And I'm going to tell you a little bit about that. This was a continuation of the first postdoc that Grace mentioned, where I was exploring feeding behavior in a couple of different populations of these fish. Especially the 3 that are listed here. I had a couple other populations, but these were the ones that I was really excited about. We've got Banfield Inlet as our ancestral marine population, and then we've got Black Lake and Frederick Lake. There's some really important differences between Black Lake and Frederick Lake, especially their elevation and how difficult they are to get to, as Anya can tell you. But I started to notice something curious about those 2 populations as well in terms of just looking at the fish. And you've maybe already caught it, which is that they look quite different from one another, right? Our Frederick Lake fish has a much bigger, bulkier body shape. It's also got a more shallow craniofacial profile. And to me, and hopefully by now to all of you, those traits were screaming benthopelagic variation to me, right? Those are benthopelagic traits that I've been telling you about all night. And that wasn't the only thing I noticed. And I don't have nice visuals to support it, unfortunately. But I brought these fish into the lab as wild-caught adults in 2019, and when I did, if I had put all of the fish into one tank, or someone else did it, and I came into the lab and they were all mixed together, and I put them into a bucket and I started sorting them out, I was pretty confident that I would be able to sort out a black lake fish from a Frederick Lake fish, no problem. They looked that different, and the differences were consistent across individuals. By the time I stopped filming them in 2020, and again, that's the same individuals about 9 months later, 9 to 10 months later, I couldn't have done that anymore. They were starting to look much more similar to each other, and specifically, the black lake fish were starting to look way more like a Frederick Lake fish. And to me, again, that screams plasticity is what's keeping these things different. And together, those two sort of anecdotal bits of evidence were making me a little bit itchy. And the reason for that is because in 2021, shortly after I finished filming my fish, Black Lake was clear cut. And as you can see, that's pretty drastically changed the face of the lake. And because Black Lake was the one that was a little bit different relative to the others, like for me, Frederick Lake looked a lot like a marine stickleback, which Dan Bolnick got really excited when I told him that. But because it was Black Lake that was the interesting and unique one, I was worried that with the clear cut, any of those differences that made it interesting and unique were going to start to go away. So as I started my position at UFE, I started coming back out here and just catching fish and then measuring their shapes year after year. I knew especially that I needed to get a clear baseline for Black Lake as quickly as I could. So I came out here for the first time again in 2023. And that's just a few generations from that logging event. And I started collecting fish from Bamfield Inlet, Black Lake, and Frederick Lake. And thanks to a very serendipitous cold contact of Katie Peichel and Dolph Schluter, I was able to get another population, which is what I was calling Oyster Bay, but apparently is Oyster Lagoon, which is a second marine population. So now I have 2 marines and 2 freshwaters. So I got all the fish, I euthanized them, I put them in tubes individually labeled, and I brought them to Friday Harbor, just across the border, to use Adam Summers' CT scanner. The reason I went there as opposed to UBC or somewhere else is because he doesn't charge me per hour. And when your scans take 7 hours, not being charged per hour is really nice. So I brought them down. It's a fun conversation at the border. We can talk about that too if you're interested. But I bring them down to Friday Harbor, I scan all the fish, and then I bring them back up here with all the data. My students typically process the scans and get them into the shape that they need to be to actually do the data analysis, and then they get to do their own projects on all the different traits. So basically, each student kind of takes a trait, and that's their data set for the year. So the first story I want to tell you about with these fish is the body shape. Following some of the work that I was doing in cichlids and zebrafish, we found that Black Lake was, just like I expected it to be, kind of off in its own world in terms of body shape. So that's confirming what I anecdotally was seeing, that Frederick Lake is clustering a little bit closer to the marines, and Black Lake is kind of out doing its own thing. And compared to those marine populations, Black Lake fish are, in fact, again, more torpedo-like. They don't look like blimps, they look like torpedoes, is kind of the way that I think about it. And Frederick Lake sort of clusters closer to the marines. When we repeated these analyses in 2024, now we're starting to see something a little bit concerning, at least if we care about Black Lake as a weirdo population, which is that it's starting to maybe overlap a little bit with our Frederick Lake population. Before they were completely distinct, now they're starting to look more similar to each other. And when I compare the shape of a fish that's out on its own in Black Lake versus the shape of the Black Lake fish that are now sharing space with Frederick Lake, again, it's subtle, but I'm starting to see some differences in terms of how bulky, how deep their bodies are. So we are starting to see these fish changes. It could be artifact, obviously. It's pretty subtle. We have a limited sample size. I'm only allowed to catch 30 fish from the freshwater populations. So it's there's that concern. There's limited amount of time since the actual disturbance event. And Actually, with 2024, we were having some issues with the scanner. It's since been replaced. We've since rescanned all of those fish and are working on reanalyzing them as well, so that might help clarify things. However, assuming it's not artifact, I started to think about what might be changing in this lake that would explain it, other than the obvious sort of nutrient wash-in from the clear cut. And one thing I have noticed in that lake since coming back, in 2019 and 2020, I was sampling the lake pretty extensively. Never once do I have a record of catching a rough-skinned newt there. Now I'm pulling them up every time I go sample. So there is a potential shift, at least in their perceived predation, because I think the newts can and do sometimes predate on the stickleback, especially the young ones. So that might be changing things. Meanwhile, in 2023, we see similar patterns of Black Lake being a weirdo and Frederick Lake clustering with the marine fish for a different trait, which is their lateral plate, their defensive armor. This follows directly from what Heather Jamnitsky's done in terms of the actual measuring of the shapes. And we're really interested, I haven't had a student pick it up until this year, but I'm really interested to see what they find about 2024, 2025, and now we have 2026 data as well. So the last thing I want to say before I move on, because my collaborator will be mad at me if I don't, is that we are also now doing, and I think some of you, at least the folks in the stickleback course, are going to come down to the lab and see what we're doing. And my student Anya is going to talk your ear off about it, I'm sure, because she's very excited. We're all excited. But what we're doing now is actually, I've teamed up with a colleague of mine who is a microbiologist, and we're trying to heat shock the fish to simulate climate change and then see how that's changing their microbiomes. And the hope is that we will be able to develop like a probiotic to help them react to and adapt to that climate change happening. So hopefully we'll have some great news on that front soon. So far we're heat shocking fish and they haven't died, so I'm pretty pleased, because that's not always easy. All right, so to wrap up this second vignette on stickleback, I have identified some populations where plasticity might be playing a role. Those are pretty crucial for my planned future, but not yet funded, genetic basis of plasticity research in stickleback so that we can compare across these very widely different Teleost bony fish. I've also got that interesting population that may be actively evolving in response to a recent disturbance, and I plan to keep following up there as we go forward to sort of document that evolution in action. And then of course we've got the microbiome project looking at rapid changes as well. So with that, I have lots of time. I'm happy to take questions on any of this. I'm also happy, given the folks who are in the room, to talk about like career paths or why I'm at a PUI and not an R1, all those kinds of things as well. So whatever you're interested in, we can chat. But I have lots and lots and lots of people to thank. Many people enabled this work, especially my students and all of my collaborators. So thanks.
So regarding fish schooling and shoaling, I am aware that fish generally prefer to school or shoal with fish that resemble them in appearance. So I'm wondering, and this might not have a clear cut answer, but if the Frederick Lake and the Black Lake sticklebacks could have some potentially have some social cue that's playing into their phenotypic plasticity.
Oh yeah, absolutely. I'm sure that is the case. Stickleback and the lakes don't school quite as often or as readily as the marine ones in my experience. At least these lakes. Can't speak to every lake, but yeah, so it's definitely, there's social elements for sure. And part of the lack of schooling might be increased parasite load in the freshwaters. Yeah. Yeah, I saw there was another hand sort of back in the same area. Yeah. All good.
You're talking about like predators. I know one of my other like professors have done something similar to this one on like the productivity of a lake.
Yeah.
Like directly affecting the shape and sizes of the spines and the plates of the stickleback. Do you think that could like possibly be done for these lakes as well as like some sort of like predator like breakdown or something to see what's like the biggest threat in those like ecosystems?
Yeah. So my ecologist husband always gets really frustrated that we don't know more about these lakes, and I think that's a really good point, to do those kinds of just sort of baseline understanding. What does the lake look like? What's the shape of it? And the other question that I routinely get is, what are they doing out in the deep water? You're catching them from the shore, so there's all sorts of things that could be going on with that for sure. I don't have those data in hand, but it's definitely a question that's been on my mind for sure. Yeah. I'm gonna go back there because you can talk to me anytime.
So, from my understanding, you were saying that you are hypothesizing that there's a big plastic role in the difference between like the Black Lake and the forest.
Frederick Lake, but yeah.
But then since the logging, you're starting to see a shift in the Black Lake population, and your theory is because of these newts. Do you think that they're causing a change in behavior that then results in also plastic shifts of those fish, or is that like not? Do you have any theories about that, or is that?
Yeah, I think the nutrient availability is one thing, and I think the predation regime is a second thing, and I think they kind of layer on and build on each other. So doing that 100% dissection of, oh, it's definitely all the nutrients or it's definitely all the predation, I don't know that that's going to be the case. I suspect it's probably both of them, and maybe, as you kind of were hinting to, they're interacting with each other as well, right? Because they're not unrelated to each other. This is all food web kind of stuff. Yeah. In the back, and then Andrew.
At the beginning of the presentation, you were talking about the variation that you see in plasticity itself. What makes you say that plasticity is costly? That's something I've understood. What can be costly about it?
Yeah, so one of the things that people talk about the most is essentially like the sensory mechanisms, having to maintain those sensory mechanisms and be mechanosensitive. And, that's one potential cost. There's also potential costs in terms of like sticking your neck out to go explore your habitat and see what it's like, right? So you have to actually be out and gathering that kind of information, which might put you at risk of exposure to predators and things like that. So yeah, the behavioral aspect of it. Yeah. Andrew.
So would it help you if we took Frederick, Banfield, and Black Lake fish and mix them all together, let them interbreed, and create a mapping cross for you?
I mean, that wouldn't hurt.
I got Frederick and Black Lake fish today, and marine fish, and that's kind of the plan for the pond over there. So.
All right. Yeah, let's do a cross. I'll never say no to a QTL. Yeah. Yeah, I was part of a joint meeting for Ix and Herps Society, like early career symposium a few years back, and that was basically everything that I talked about. Was essentially like fish are super plastic, and it makes them very evolvable. And so that's one of the reasons I think we have so many fish species, is because they are so plastic and they can sort of use that as a springboard for evolution. Does that answer your question? Yeah. Yeah, just fish in general seem to be very plastic. Yeah.
So some of the marine fishes are the ancestral population, and then they came to the lakes. How did they get to the lakes?
Yeah, all right, so let's talk about a tsunami. So one of the reasons why Dan got so excited when I started telling him that I was seeing morphologically that the Frederick Lake fish were more similar to the marines was that he had done the genetics already and found that they are more closely related to the marines. And we think what happened there, Frederick Lake is super low lying and it's quite in the way of any potential tsunami, whereas Black Lake is a much higher elevation to start. And so there's a pretty well documented tsunami that hit the area about 400 years ago, and we think that's what washed those fish into that lake. How they got into Black Lake, either they were there before the elevation was so high, or there's all sorts of mechanisms, including things like fish swimming up little inlet, little sort of river channels. There's some theories that they can be brought in by bald eagles. You know, we know some stickleheads who like to just throw them in lakes. So there's all sorts of ways they could have gotten in there, but it was probably something along those lines of like being carried in.
I was wondering, so how is it then that they can move from a marine to like a freshwater environment? Like the different osmotic, I don't know what to say.
Plasticity. To be honest with you, right? It's just plasticity in a physiological trait as opposed to a behavioral or a morphological trait, but they are highly plastic. And stickleback, at least some of them seem to be anadromous like salmon, so they come up to spawn, so that's part of their system already. Yeah. Yeah.
Are there any mechanisms that we have now that can like measure these like upregulations of genes that are causing this like these plastic physiological signs that we can see? Like, because it must be, it can't just be like nothing going on other than, oh, if we're in a new situation, it like just becomes that way. And it's still sensitive.
Yeah, I mean, I like to take a broad net, which is why I was going for RNA-seq and ATAC-seq in my postdoc, because it gives you sort of the whole picture. You get all the data. Not all of it's meaningful, but you get all of it. Whereas with the QTL, it's a little bit more targeted and precise, which is amazing for other reasons, but I kind of like to have all the tools in my toolkit if I can. And I think those are some of the ones that I think can get at the questions that you're talking about. Yeah.
So you're trying to like measure plasticity.
Yeah.
Like, are there, like, do you think that there are different populations of stickleback that are like more or less plastic?
Yeah, I had a really interesting conversation with Heather because she and Sean did some work on the marine populations, and it seems like there's maybe more population substructuring, right? The predominant theory is like, oh yeah, they're all one population in the marines, but how could that be possible, right? They're not always all interacting with each other, so there's probably some substructuring there. I think the same is true within a population in a single lake as well, right? We might go out into the deep water and find that they're doing something completely different out there than they are in the near shore sort of subpopulation, I guess. So I do think there's those kinds of differences going on for sure. Whether I have the money, time, and energy to go after them is another question, but yeah. Yeah.
Thanks for your talk. It was really great. I was just wondering when we were talking about the plasticity bone density in the stiffened MTF.
Less density, more deposition. So how much bone are they laying down? Yeah, yeah.
Is it like new bone deposition, or are they like taking minerals from elsewhere to bring it up to the head structure, or are they
Ooh. Oh. Well, okay. So I think I didn't really think about that before, but I do think with fish, they're constantly laying down new bone. So it's not really, right, they're sort of continuous growers. You put a goldfish in a small tank, it's not going to grow very much. You throw the same goldfish into a big pond, it gets bigger and bigger and bigger, right? So they have somewhat continuous growth, which means they're constantly laying down new bone to support that growth. However, the idea that it could be taken or shunted from other traits into the head, I think that's a really neat idea. I'm not. Yes, exactly. Yeah, and we tried to do some, Joseph, one of my first students at UFE, tried to do some calcium, but we didn't actually get any viable calcium data. So we kind of abandoned that and just looked at marine versus freshwater, yay. But I think that's definitely something I'd like to do in the future for sure. Yeah, knowing the ecology of the system is really important. Yeah.
More of a comment, but instead of being your science communicator, like this presentation was super well put together.
Thank you.
Maybe if it can up your biology or starting, try and put a lot more effort into teaching how to make a good presentation. It's not easy. So I think like your use of colors and arrows and all the tricks there is really good. So undergraduates, we get to make a presentation like this is exactly what a rocking presentation is.
Thank you. Really appreciate that. Happy to guest lecture for SciComm at any time. It's been a main focus of mine for a long time. So, yeah. Yeah.
Thank you so much for your talk this evening. I love to ask this of all of what I call fancy people who get to come here, meaning
Me and my
people who've gone through a very extensive education level, are now often speaking as experts in the word like you are now. And many folks in our summer seminar series
what is something like, what's a job that you had or a stint of time that you spent that is like not even related to what you're doing at all that you had in your path?
Oh, see, that's tough because even the jobs that I had, I realized in hindsight, and I just wrote this in my tenure application, so it's all fresh for me. But like, I was a camp counselor and I was a sailing instructor for basically my summer job in high school and college. But again, how much of that is unrelated when like I use the knots that I learned through that experience? I talk to students just the same way I talk to my campers. Well, not just the same way, but but you know, those skills were part of that process. So I'm not sure that. I mean, I was a lifeguard for a while. That's probably the least related job that I had. And I still have first aid training for being out here because that's important to me to know that, you know, if my students get hurt, I can help them. So again, I don't know, it all seems really related in hindsight. And I will say that that's kind of a privileged take because I didn't necessarily work retail as an undergrad and things like that. So that might just be unique to me. So I want to acknowledge that as well. Yeah.
Any other questions before we wrap up?
Ooh, yeah. Two-part question. Yeah, okay. Yes, it is difficult to get funding. Period, point blank. That is the time that we are in. There's no point sugarcoating it, right? So that's unfortunate for sure. But I don't want to leave you walking away from here going, there's no hope whatsoever, right? Like I found a way. Again, I've gotten very lucky both in my mentors and in just my life experiences that I got lucky that way. But it's certainly feasible to continue going for academic jobs. You know, it's just a little bit harder to argue for funding for basic research nowadays. But all the more reason, as you said, to have stronger K-12 education programs, to have stronger science communication as part of our backgrounds as academics. And I'm starting to see that change. When I was in grad school, there were several faculty members that I interacted with who treated their grad students who were interested in pursuing science communication like it was a joke. It was a washout career that you got because you couldn't do academia. And I think that perception is changing. I have seen that perception change over the last decade. So I do think we are starting to value that, at least within the community. I don't know how much we value that as a general population, but hopefully the pendulum will swing back eventually, especially if we can keep investing in education and SciComm. Yeah.
You mentioned how many more collaborators you're experiencing now. Have you found over the course of your career that science in general has become a much more collaborative like way of going now, or is it just now more recognized how many more people are a part of these processes?
Yes. No, so I think on the one hand, I've been very careful with who I collaborate with, and because of that, I've been able to develop have their networks become my networks. And so the people that my PhD supervisor introduced me to are some of the people I'm still collaborating with today. And then you just sort of build those networks as you go, right? I do think folks are realizing that we can't all know everything and that we need people to be experts in one or two things, and then we get together with experts in two or three other things, and then we have a really cool project. So I do think the perception's changing. I also think that it's worth being intentional about those relationships that you're cultivating. Yeah.
Any other questions before we wrap up?
I will also say I'm happy to grab a coffee and chat anytime. Feel free to just reach out. I think my email's up there somewhere. If not, it's dina.navon@ufv.ca. Not super hard. Dina.Navon@UFV, University of the Fraser Valley.ca. That's true. Yeah, you can Google me.